Hi, Nicola. Hi, everybody. Great pleasure to be here with you. And obviously, quite a number of you will be listening to this online in your own time, which actually works reasonably well. Nonetheless, I hope to see you all in 3D sooner rather than later. So, the topic that I was asked to cover today was what we have learned from birth cohort studies, and I'm going to focus on asthma across the life course. I'm not going to be talking only about birth cohort studies because there are some tremendously important studies which have been done in a context of cross-sectional and other types of studies. So I'm going to start by focusing on the key question. What are the advantages of birth cohort studies? Why do we need to know about life course trajectories rather than rely on the data from cross-sectional studies? And I'm going to start by giving you a really simple example, asking a really simple question: what is the role of exposure to domestic pets and inhalant allergens in the development of sensitization and subsequently asthma? How can we understand the complex relationship between exposures and genetic predisposition? In the context of birth cohort studies, an increasingly large amount of data that we are collecting, can big data and modern techniques help us understand the disease and ultimately be translated into clinical practice? I'm going to talk about, terms that are buzzwords now, like artificial intelligence and machine learning. Can we use machine learning to unravel the complexity of asthma and allergies and their relationship? I would like to take you to really different concepts and take you beyond atopy and allergic sensitization and discuss the role and relevance of different patterns of sensitization in relation to asthma. Ask a question as to whether severe asthma is a separate subtype of asthma, and can we understand asthma by focusing on this sort of extreme end of spectrum. And I'm going to talk about various complex methodologies to address the longitudinal nature of data and try to understand how it relates to clinic. I'm going to use two examples, one which I'm particularly interested in, which is trajectories of lung function from preschool age into adulthood, and what's the relevance of early life exposures on lifelong health. Talk about trajectories of exacerbations and their patterns in relation to asthma severity. So let's go to the beginning. Let's discuss this key element, really important element of time. Let's compare the data from cross-sectional studies and birth cohort studies taking into account life course trajectories. And to use this, I'm going to use a very simple exemplar. Is cat or dog ownership in early life good or bad for you? And for all of you who have been at various meetings, both respiratory and allergy, you will know that pretty much at every meeting, there will be a pro-con section. Cat ownership is good for you or bad for you. And to be quite honest, I have taken part in a number of such debates, being on both sides of the argument. And being on both sides of the argument is not too difficult because there are fantastic studies showing that having cat in early life is protective, i.e. that it's good for you. There are equally good studies showing exactly the opposite, that actually having cat in early life is really a very strong risk factor for subsequent disease or allergic sensitization. And then a third group of studies showing, well, quite frankly, having cat, not having cat doesn't really matter. Who cares? Doesn't make any difference. Now, this is an area full of controversies and conundrums that has been sort of going on for best part of 2 decades. And the question is, when you look at these data, first one is, which one of these studies is really poor and we cannot trust it? Well, looking at the study design, they all seem to be pretty good. So is it possible to explain what are clearly, apparently, completely opposite results? And there's also really fascinating question hidden behind this, which is, what if all of this is true? So how can we address this question? Well, to do it, we ask a very simple question within the context of birth cohort study. What is the pattern of the development of cat-specific sensitization through childhood amongst children who had cat in an early life and those who did not have it? It was done by Hilton Ihuma, really great fifth-year medical student. And within Manchester Asthma and Allergy Study, our birth cohort, we had the information on cat ownership in early life and cat sensitization throughout childhood. And to start with, we used very, very, very simple descriptive statistics, asking the question, what is the proportion of children with allergic sensitization to cat throughout childhood who had or didn't have cat in early life? And it is very clear that proportion of sensitized children here in black is markedly higher amongst children who lived in a home with a cat in early life, but only to age 3 years and not thereafter. And this association between early life cat ownership and exposure to cat allergen, which we can measure in a home, really changes fundamentally during childhood. And the key factor that will determine the conclusions of your study is time when you assess the outcome. So if you assess outcome, which in this case is sensitization in preschool age, the inevitable conclusion is having cat is really bad for you. If you do it during school age, well, having cat or not having cat will really have precious little effect on the outcome. And we now have the data in adulthood, and in adulthood, it's really clear that the situation reverses. So indeed, depending on the timing of the assessments of the outcome, your study can come up to all three conclusions. Cat is risk factor, cat doesn't matter, cat is protective. Fundamentally different conclusions. And the true difference is really in the trajectory of the development of clinical outcome, which in this case is sensitization, in relation to early life exposure. And what you can see is that amongst children who have been born with a cat, this is really flat curve, i.e., whatever happens, happens really early, usually by first or third years of life. Amongst children who live in a home without a cat, the slope is fairly steep, and by adolescence, those curves actually cross, and the proportion of individuals who develop sensitization in the group of those who did not live in a home with a cat actually exceeds those who live in a home with a cat. So unless you look at trajectory, you will inevitably come to sometimes very misleading conclusions. So indeed, cat ownership can be risk, can be protective, and will not matter depending on the timing of the assessment of your outcome. And the only way you can understand the true relationship is by mapping lifelong trajectory. Now that is what you can do only within the context of birth cohort study. Now you can ask question, well, okay, how about the impact of genetic predisposition? How does that change this complicated temporal relationship? And you can think about, well, which genes should we pick up? And the good one is filaggrin. Most of you should know about filaggrin. It's a major risk factor for eczema and for various dry skin conditions. And filaggrin loss of function mutations result in higher permeability of skin. So skin as a barrier is really a little bit leaky amongst individuals with filaggrin loss of function mutations. That's why they have dry skin, ichthyosis, develop eczema. But when you think about it, if skin is more permeable, then you can speculate that impact of any transcutaneous exposure may be amplified amongst individuals with filaggrin loss of function mutations. So very recently, we carried on to do much more complex analysis, asking question, what is the impact of early life inhalant allergen exposure, which we measured at home, how much house dust mite allergen there is, how much cat allergen is, how much dog allergen there is, in relation to these specific sensitizations amongst children with and without loss of function mutation. And when you do longitudinal modeling to understand it, it becomes really interesting. For example, in relation to house dust mite exposure. Exposure to house dust mite allergen increases the risk of sensitization. This is markedly amplified in children with filaggrin mutations, but only in early life, very early. So these very early sensitizations to inhalant allergens, children who are particularly susceptible to the impact of exposure are those with filaggrin loss of function mutation. And this gene by environment interaction then gradually diminishes as children grow older. So it is very clear that genetics, in this case filaggrin, modifies the relationship between exposure and sensitization. But the effects are fundamentally different at different ages. And they also differ between different allergens. So all of this work, in a nutshell, tells you that if you have a child with filaggrin loss of function mutation, probably the best thing you can do for them is make sure that they have a dog in their home, because this seems to really impact upon development of their immune system, probably via exposure to microbial agents which are associated with dog ownership. They should stay away from a pet, and probably a reasonable thing in this group of individuals would be to minimize inhalant allergen exposure in early life. What is also very clear is that if you're trying to understand the impact of both genotype and exposure, the timing when you assess your outcome will be critically important and will have impact on the results. Now think about it. You know, people are currently really insisting that you should replicate all of your findings, particularly in relation to genetics. What I would say is replication is rather a dangerous word here, because the only way to replicate is if you have absolutely identical study design and identical age of the assessment of the outcome. To understand what is going on, to understand these complex relationships, first of all in terms of disease development, but also the complex relationship between genes and environment, you need to look at life course trajectories. And that, unfortunately, is rather tricky, both in terms of collecting the longitudinal data, but also in terms of the analysis of this data. So what are the challenges for the analysis? Well, the challenges are that, you know, we are now capable to collect vast amounts of data very quickly. You know, you have high throughput technologies. You know, we are collecting now terabytes of data, and it's becoming increasingly challenging to process, analyze, and interpret this data. And sometimes, you know, this vast quantity of data makes it impossible to say, well, what is actually the question that I'm asking for? You will all have witnessed this shift from standard scientific method of traditional hypothesis-based research, where you have very carefully constructed questions and observation, towards data-driven, hypothesis-generating approaches, unbiased approaches, and these are now overshadowing traditional approach. Being myself in sort of field of machine learning and AI for best part of last 20 years, I find it really interesting that big data now are being sold to us as panacea for generating hypothesis and driving new frontiers of healthcare. It is data that will speak for itself and that will solve all the problems of the world. And people talk about AI, machine learning, deep learning, systems biology, very often using these terms interchangeably. And let me make it crystal clear, interchangeable they're not. So let me give you an example of how you can use these modern techniques of machine learning to understand disease, and whether there is any way we can translate it into improvement in care in day-to-day clinical work. I will start by giving you just a sort of taste of what machine learning is. Generally speaking, in computer sciences, in gaming, they very often use predictive machine learning. What is predictive machine learning? You want to make prediction. You want to be fairly certain to be able to make relatively accurate prediction of an outcome from the data that you have, but not necessarily to understand why this data and the algorithm actually predicts the outcome. For me, for medicine, explanatory machine learning is much more intuitive, because the aim is not only to predict something, but also to understand the complex patterns in the data. And I think it's really well suited for medical research, where there's a human in the loop, and human wishes to understand the process that gives rise to the data and the outcome. I'm going to give you an example of explanatory machine learning in taking you beyond data. Now, you will all know that, there are sensitized children out there. So let me ask you a very simple question. What is sensitization? Sensitized child is the one with positive skin test or positive IgE. Well, come on. You all know that amongst patients you have seen in the clinic, there's huge heterogeneity in terms of the patterns of their skin test responses and their IgEs, size of response, allergens that they sensitize to. And yet, we call child atopic if they have one positive skin prick test, let's say to house dust mite with mean wheel diameter of 3 millimeters, or if they have large number of positive skin tests with very, very big mean wheel diameters. And you have to ask question, well, actually, are these different sensitizations exactly the same? And how are they related to asthma? Now, to do this, we collaborated with people in Microsoft Research Cambridge, and this is really one of the first examples of the use of machine learning, certainly in allergy and respiratory medicine, probably in medicine as a whole. And the question was very simple. Can we understand this feature that we call sensitization a little bit better by looking at the patterns of the development of sensitization throughout childhood, taking into account different allergens measured by skin test and specific IgE at different ages, but at the same time, taking into account existing knowledge. Because black box approach, I think, is very silly. You throw everything into a model and hope for the best. But actually, we know a lot about it. We know that child sensitized to egg at age 1 and 3 is at higher risk of developing subsequent dust mite sensitization. We also know that if you have large mean wheel diameter in early life, you're much more likely to retain this sensitization than to lose it. So you can build a model that is based on all the data that you can have, plus the knowledge from previous studies to try to understand the patterns. So think about it. Here, you actually, for each child, have skin tests to 8 different allergens, IgEs to 8 different allergens, at 4, then later 5, 6 time points. This vast amount of data that is very difficult to look at and see the pattern for human. But you can teach the machine to run the algorithms and understand the pattern. And what this has told us was that this idea that there is such a thing as sensitization is fundamentally flawed, because amongst children who we would traditionally consider to be sensitized, there were at least 4 different types of sensitization, which is interesting. When it becomes much more interesting is when you look at the association between this different subtype of sensitization with asthma here. These are 2 birth cohorts with strikingly similar findings, and that is only 1 subtype of sensitization is associated with asthma. Amongst children who are sensitized, only a quarter actually fall within this very specific subtype. But the strength of the association with asthma is staggeringly high. They're 20 to 30 times more likely to have asthma. And not only that, when we look at all the important outcomes of asthma, like lung function, exacerbations, and hospital admissions, it was children within this specific subtype of sensitization that were at much higher risk of having all the markers of asthma severity. So it is clear that what we currently call sensitization actually comprises of several different subtypes. Some of them are completely benign and irrelevant in relation to asthma. Some are clearly important and pathologic. And it's really great because if I run a birth cohort study and if I have vast amount of information over long periods of time in thousands of children and I model it, then I can assign each of these individuals to different subtypes of sensitization. But that's not what happens when I'm in a clinic. In the clinic, I have 1 shot at it. So is there any way to try to determine amongst sensitized children who are the ones with this pathologic subtype of sensitization, with a subtype of sensitization that matters and that we should take into account? Now, clearly, we cannot do it based on these standard measures like IgE or skin test to whole allergen extract. But there has been really tremendous change in the last 5 to 10 years in our technical ability to measure this feature that we call sensitization. So currently in clinical practice, you have what is called component resolved diagnostic chips. So rather than measuring IgE to house dust mite or to cat or to peanut, these chips measure IgE to individual allergenic proteins from large number of sources. So there will be several allergenic dust mite molecules, cat, dog, pollen, food. But what you get is a readout on 8 pages listing 112 different results. Now, how do you interpret this? How do you make sense out of this? So we ask the question whether this complexity is actually a good foundation to try to understand whether we can differentiate between pathologic and benign sensitization, and whether we can use now predictive machine learning, whether we can just put this data for each individual, 112 different IgEs, into the model, and then the model can differentiate between benign and pathologic, and whether this can then predict presence of asthma. And it was quite striking that the patterns of interaction on a chip among sensitized individuals were very strongly predictive of asthma. Now, for all of you who have looked into various algorithms for asthma diagnosis. This is quite striking because among sensitized individuals, just by running the model on the patterns of IgEs in these complex chips, without asking any other questions, we can predict asthma by area under the curve of 0.94, which is quite spectacular. How and why does it happen? We don't understand, but it is really a nice example of predictive modeling. You sit in a clinic, you say, well, the question that I have is, A, does this child have asthma? And B, is this sensitization relevant to asthma? We're almost there to be able to do it. Can it predict asthma severity? Now, this is the work that is really, really interesting and is now taken into case control study, famous BioPred study, together with Graham Roberts and Louise Fleming. We looked into the patterns of interaction on these component resolved chips in relation to asthma severity, to see that the networks of sensitization to individual molecules is completely different in severe and mild moderate asthma. To the point that we can now pretty much say, in a sensitized child with asthma, whether their asthma is genuinely severe or not, just based on pattern of sensitization. With network in severe asthma having really, really high connectivity, you see there's a lot of connectivity between IgEs, but these individual connections are not as strong as in mild asthma. You see there are much darker connections. So it is also fascinating, I think, from the point of view of trying to understand what drives severe asthma is that the connectivity between IgE to different molecules exists for proteins with no structural homology in severe asthma, whilst in mild moderate asthma, the connectivity is really strong for structurally homologous components. What it tells to me is that mild asthma is much more of an IgE-driven Th2 process, whilst there is an interesting dysregulation of responses that is probably epithelial that exists in severe asthma. But this is pure speculation. So what about severe asthma? Is it separate type of asthma? So again, this is now use of top-down, unsupervised, and supervised learning models brought together, complex mathematics, looking in more than 600 exquisitely well-defined asthmatic children, where by using 4 different domains, time of onset of symptoms, whether they were sensitized, what was the pattern of their severity and their recent exacerbations, uncovering 5 different subtypes of asthma. And they will be kind of familiar to you. Early onset atopic asthma, early onset non-atopic asthma, late onset asthma, difficult asthma, exacerbation-prone asthma. For all of those who have seen these clinics, these will be very, very, very familiar. We observe it in a clinical practice, but here we can genuinely see the differences based on the patterns of clinical features. It will not come as a surprise to you that children with difficult asthma have diminished lung function, that blood eosinophils are present in difficult, early onset mild atopic and in late onset asthma, but not necessarily in some other forms. But where is severe asthma in all of this? Well, what is really interesting is that severe asthma is present in each one of these different asthma clusters. What it tells you is that there are a number of different pathways towards severity, and that severe asthma is just extreme end of spectrum of a number of different asthma endotypes or clusters. Of course, the relative contribution will be different in that generally there will be higher proportion of severe asthmatics amongst those in difficult asthma cluster, but there are severe asthmatics in all of these subtypes. So really, to understand asthma complexity, you need to look across all different ranges of severity. Now, what can we do to understand the disease and to capitalize on longitudinal data? Because doing simple models is clearly not give you the satisfactory answer. So can we bring together the complex analysis and capitalize on longitudinal data? So I'm going to show you a few examples where latent variable modeling, and latent per se means unobserved. So these are the patterns that you cannot observe, but you can uncover, and that can tell you something important about the disease. And the first one is that of lung function trajectories. So this is the work that we've done in 2 birth cohorts, Manchester and Alspach. Initial modeling in about 2,000 individuals with repeated measures of lung function from early school age, from age 8 years, through school age, into adolescence, and then early adulthood. Why is early adulthood important? Because, in 3rd decade of life, you reach physiological peak of lung function. Now, by modeling lung function, we have observed in both birth cohorts strikingly similar, almost identical patterns. In that, about 4% of population individuals will have persistently high lung function, super good lung function. But there's also 3 to 7% who have persistently low lung function throughout childhood, from preschool age into physiological peak. And not only that, we can then do further model to understand what are the predictors of persistently low lung function. And persistently low lung function throughout childhood is predicted by 2 features: multiple early sensitization, the one that I was just talking about, this interesting subtype of population of sensitization that is strongly associated with asthma, and recurrent wheezing with severe exacerbation within first 3 years of life. So key feature, severe troublesome early childhood wheeze with severe exacerbations. Remember this and park it. We have done these analyses together with, and we tie in publication with our colleagues in Tasmania, who model lung function now from childhood well into adulthood to 6th decade of life, and described pretty much identical patterns. The ones that we couldn't obviously observe due to nature of very, very careful data collection in early life, but really little data past age 20, is this group of individuals which catch up with lung function. And we have shown subsequently that yes, this does exist. And, those individuals who have below average lung function and then accelerated decline, which probably is real disaster. But what was very interesting was that they had an opportunity to look at COPD in 6th decade of life. And COPD in 6th decade of life was predicted not by whether they started smoking in their 30s, but with poor lung function pathways in childhood. And 3/4 of COPD begins with poor lung function pathway in childhood. COPD is genuinely a disease with early life origins. And we need to understand it, because if we are to change it, it is far too late to do it in the adulthood and among smokers. And childhood characteristics that predict lung function trajectories and COPD are childhood asthma, particularly severe asthma, bronchitis, rhinitis, pneumonia, and heavy smoking mother. Interesting, isn't it? So the conclusion from all of these is it's really important to pay attention to early life events. What then emphasized the tremendous importance of this is this analysis by Alvaro Agusti in 3 large cohorts, that's Framingham, Offspring, Cardia, and Generation 3. in which he looked at the relationship between lung function at physiological peak, you're looking at 25 to 30 years, with subsequent respiratory, cardiovascular, and metabolic outcomes. Now, perhaps by what you have seen, it is not surprising that he has found that FEV1 below 80% at physiological peak in third decade of life increases at subsequent risk of respiratory disorders and COPD. But what was really interesting was that the diminished lung function that clearly has origins in early life is also associated with much higher prevalence and earlier onset of cardiovascular and metabolic abnormalities. And there was a higher and earlier incidence of comorbidities brought forward by about a decade in those with diminished lung function at physiological peak. Not only that, lung function at physiological peak is one of the strongest predictors of all-cause early mortality. And all of this is completely independent of, but additive to cumulative smoking exposure. Now, lung function throughout childhood is clearly tremendously important. And we have shown that there are features that are associated with decline of lung function during childhood. Now here, we're not doing spirometry. This is measurement of specific airways resistance in body box. And what you can see clearly is there is a subgroup of children, again, characterized by the presence of multiple early atopy and severe wheeze with exacerbations in first 3 years of life that is associated with loss of lung function throughout childhood. So we really have an opportunity to do something about it in early life. The features are multiple early sensitization, persistent wheeze, severe exacerbations. So why exacerbations? What are we looking there? Is this strong? What's the pattern of exacerbation? You know, when you talk about exacerbation, we all know clinically that there are frequent exacerbators. It's really difficult to identify them without being very, very, very subjective. So to ask this question, we modeled severe exacerbations in a birth cohort where we transcribed all of the healthcare records and had information about acute severe exacerbations as defined by ATS and ERS throughout first 8 years of life. And we then again applied machine learning methodologies to see whether there are any specific patterns. And indeed, amongst children who wheeze, 60 to 65% do not have exacerbation. So about a third of early childhood wheezers have exacerbations as feature of their disease. But there are two very different types or clusters. One is infrequent exacerbations, but really interesting, very small subgroup, about 10% of wheezers, with a pattern that we called early onset frequent exacerbations. Look at it here in the magenta. They are characterized by quite a large number of exacerbations, start early, etc. You may ask, so what? So what? Well, so what comes when you bring all of these individuals and then follow them for another 10 years. So to do this study, it takes about 25 to 30 years of your commitment, but you can then map their lung function in relation to patterns of exacerbation. And it's fascinating, isn't it? The children without wheezing, those who wheeze but without exacerbation, those with infrequent exacerbation, they're almost indistinguishable in terms of their lung function from mid-childhood into adolescence, and now we have the data at physiological peak. It is those individuals with early onset, frequent exacerbation that progressively lose lung function. And you will have all seen what the functional consequences of this are. It raises really staggeringly important questions. Should we start measuring lung function in children as they get into school? If so, what on earth can we do about it? But it would appear that lung function in childhood is really important and excellent predictor of troubles to come. So, birth cohorts are tremendously important because life course trajectories are telling us so much to understand the complexity of human disease. Now, of course, while informative, they cannot provide you all of the answers because you desperately need patient cohorts, particularly severe one like Sejal's severe asthma or Louise's, severe preschool Louise's or Louise's severe asthma. Because even in huge birth cohorts, there are very, very, very few individuals with severe disease. You need to bring this together. The only type of study that can give you really good information about causality is randomized controlled trial. You need to commit to very careful phenotyping. You need then to capitalize on the developments in data analysis and computer sciences, apply all of these complex methodologies to really high dimensional data that you have collected. And you may get really important clues. Now, all of that will have to be underpinned by very careful mechanistic studies in mouse experiments and in human tissues. So no single data source, no single study can uncover complex mechanisms underpinning complex, heterogeneous diseases like asthma, I would say like arthritis, like diabetes. The problem is that unless you understand mechanisms, you will never be able to apply mechanism-based treatment. Now, mechanism-based treatment is actually personalized treatment. So without understanding mechanisms, there is no personalized medicine. And really, I would like to be able to know precisely why I'm prescribing individual drug to an individual patient before I'm retired. There's really urgent need to integrate all of this, all of the steps in a research process. And to do that, we cannot continue the way we're doing. We need proper cross-disciplinary cutting-edge analytics, which is based on large, complex, integrated data set. We need teams of people who can understand each other and work together. You know, we need teams, we need human expertise, we need underpinning technology and technological advances. And I will give you a wonderful example of the most complex machine that humans have ever built, Large Hadron Collider. And you know, Large Hadron Collider, without a global network of scientists who are dedicated to pursuit of knowledge, would just be a drab underground tunnel, nothing more than, you know, disused tube tunnels under London. It is this integration of technology with human expertise and different types of expertise that will help you understand complexity of biology and human disease. And with that, you know, these are all people who have contributed to generation of this data. I am eternally grateful to my friends and colleagues in Stellar Birth Cohort Consortium of 5 UK birth cohorts in Manchester, Bristol, Aberdeen, at Imperial and in Southampton. Our friends and colleagues in computer scientists who are developing methodologies, our data science teams across Imperial and Bristol, who are really working very, very, very hard, not all in terms of applying complex methodologies, but also working further of them to make them fit for the use. All the people who work then on mechanistic models, from Pat Holt, Seb, our colleagues across the world in US, across EU, only through bringing it all together will we be able to really change the outcomes. And with that, I'm happy to answer any questions that you may have.